Hello, and welcome to the Electrolyte Club. My name is Helbert Rondon, and I am a nephrologist at the University of Pittsburgh. This is lecture number 2, Basic Physiology of Water and Plasma Tonicity Homeostasis. The learning objectives for this lecture are number 1, to explain how changes in free water affect plasma tonicity and plasma sodium concentration. Number 2, to describe the clinical manifestations of free water deficit and excess. Number 3, to describe the homeostatic response to free water deficit and excess. And number 4, to distinguish between sodium and water disorders. We all have certain amount of water in our body, certain volume of water. I'm going to represent this as TBW, or total body water. And the proportion of the weight that is water varies in men and women. For men, it's about 60% of their weight. For women, it's about 50% of their weight. And as people ages, over age of 60, the proportion of the weight that is water goes down. So after age 60 years old, the proportion of water goes down by approximately 5%. So for a typical 70 kilogram man, 60% of his body weight will be water, so there is 60% of 70 kilograms, that's 42 liters. So what would happen if this man would lose 1 liter of total body water? So his total body water will go from 42 to 41 liters. But in the context of secretory diarrhea, what would be the clinical manifestations for this? So secretory diarrhea is isotonic. So this man is effectively losing water with sodium, so isotonic fluid. So if we remember the normal total body sodium and total body water maintain a normal proportion, and in this man who is losing isotonic fluid, he's not only losing water, but he's also losing sodium. Therefore, both sodium and water will go down, and this man will develop hypokalemia because total body sodium is down, as we review on the first lecture. But the normal proportion between sodium and water will be maintained, and no other problems will occur. Let's look at another scenario where we have a similar 70 kilogram man with 42 liters of water, and he also loses a liter of total body water. So his total body water goes down from 42 to 41, but now in the setting of being lost in the desert and having no access to water. So when you're in the desert, you're losing water to stool and skin, so-called insensible losses, and if you're not able to replete those losses, then a water deficit will establish. So the other issue is that these losses have very little amount of solutes on it. So for practical purposes, this man is losing pure water, or what we call free water. So this man is going to lose 1 liter of free water. What are the clinical manifestations for this? So total body sodium, total body water, again, initially normal and maintaining a normal proportion. But now, because he's losing only water without any sodium, total body water will go down. And this will not have any effect on volume status because this man is not losing sodium. He has to lose sodium to affect the volume status significantly. So this clinical volume will be around normal, but because the normal proportion within total body sodium and total body water is altered, then some consequences will occur. Now, will this man present with any symptoms or signs? Perhaps, but they are not very reliable. The best way to uncover this problem would be by checking plasma osmolarity, which will be high, and plasma sodium concentration, which also will be high. This is called hypertonicity. So when a person loses pure water, volume status remains about the same, but there is a hypertonicity that occurs. If the person loses both sodium and water, so isotonic fluid, then there is no problem with hypertonicity, but hypokalemia will occur. Now, if you are still skeptical about the effects of losing free water on volume, let's examine what happens when we lose a liter of free water. What will happen to intravascular volume? So we have a typical 70 kilogram man with 42 liters of water, and of those 42 liters, 2/3 of those will be intracellular, located in the intracellular fluid compartment, so that will be 2/3 of 42 is 28 liters, and 1/3 will be in the extracellular fluid compartment, so that will be 1/3 of 42 liters is 14 liters. Of those 14 liters in the extracellular, 3/4 will be in the interstitial fluid compartment, so that will be 10.5 liters, and 1/4 of those 14 liters will be in the intravascular fluid compartment, that will be 3.5 liters. If you lose a liter of free water from your total body water, your total body water will go down from 42 to 41, and that liter has to come from all the compartments that make the total body water and in the same normal proportion. So that liter, 2/3 of that liter will come from the intracellular fluid compartment. So the intracellular fluid compartment will lose 2/3 of 1 liter, which is 0.66. So 28 minus 0.66 is 27.33. 1/3 of that liter will come from the extracellular fluid compartment. So that will be 0.33. So 14 liters initial minus 0.33, that is 13.66. Now that third of a liter is going to come from the interstitial compartment, so 3/4 of that third will come from the interstitial compartment, so that will be 3/4 of 0.33 is 0.25, so 10.5 minus 0.25 is 10.25 liters that is left, and in the intravascular compartment, 1/4 of 1/3 of a liter will come from it, so it will be 1/4 of 0.33, that is 0.08 liters. So from 3.5 it will go down to 3.42. So this means that for every liter of free water that is coming from the total body water, only 0.08 liters are coming from the intravascular. That is 80 cc's of fluid. That is clinically insignificant and will not cause any effects on the volume status. If you would lose water with salt, then it will have a much bigger impact, but pure water doesn't affect the volume status. Alright, the normal proportion of between sodium and water that we talk about briefly on the lecture number 1 and also in this lecture, where is that coming from? Well, it comes from the Edelman equation. So Isidor Edelman was a renal physiologist that did some important research in the 1950s, and he basically had these patients with different conditions: heart failure, cirrhosis, adrenal insufficiency, nephrotic syndrome, kidney disease, and he measured physiological parameters in these patients, including plasma sodium concentration, total body sodium, total body potassium, total body water, and he found that there was a very good correlation between plasma sodium concentration and the ratio between the total body solutes, so sodium and potassium, and total body water. So this is where the Edelman equation comes from, that says that plasma sodium concentration is equal to total body sodium plus total body potassium divided by total body water. And this is called the master Dysnatremia equation, because all the other equations that you use, for instance the free water deficit equation, they are derived from this equation. So you look at the equation and you ask yourself, well, I understand why total body sodium and total body water would affect plasma sodium, but what does potassium has anything to do with plasma sodium concentration? Well, if you think about it, sodium is the main osmol in the extracellular fluid, but potassium is the main osmol in the intracellular fluid, and therefore changes in potassium could affect water shifts and therefore change the plasma sodium concentration. As a matter of fact, if you look at the equation, if your total body potassium goes down, manifested as hypokalemia, the plasma sodium concentration will go down as well. The so-called hypokalemia induced hyponatremia. How does this work? Well, you have your intracellular fluid compartment and your extracellular fluid compartment, and your extracellular fluid compartment has sodium as the main osmol, the intracellular fluid has potassium as the main osmol. And if this is because there is a sodium-potassium ATPase pump that pumps sodium out of the cell and potassium inside the cell. If you lose potassium from the ECF because of diarrhea, for instance, then the potassium in the ICF, in the intracellular, will come out to compensate for the loss. And the problem is that you're losing a cation, positive charge. So in order to maintain electrolyte neutrality, the cell has to gain a positive charge. So the cell is going to gain sodium. So sodium goes inside the cell, and therefore that will reduce the concentration of sodium in the ECF, causing hyponatremia. Well, if this process gets reversed, what happens? So if you give potassium to this patient, then the potassium will get into the intracellular fluid, and then the sodium that was sequestered there will come out and increase the plasma sodium concentration. So you can increase plasma sodium concentration by giving just potassium. And there are situations where this could have significant consequences. So people who present with severe hyponatremia and hypokalemia at the same time, you could give them potassium-containing solutions to replace the potassium without giving them any sodium, and that will make the sodium concentration in the plasma to increase. But not only that, if you're not careful, it can overcorrect. And like in this case that I'm showing you here, reported in American Journal of Kidney Diseases in 2010, this patient developed overcorrection of plasma sodium and osmotic demyelination syndrome just by giving potassium without any sodium. So do not give potassium unless the patient has symptomatic hypokalemia, and wait, if you can, until the sodium concentration is to a level that is more safer before you give potassium. So for didactic purposes, we're going to forget of the contributions of potassium to plasma sodium concentration, although you know this is very important. And we're going to say that the plasma sodium concentration is the total body sodium divided by total body water. And I'm also showing you a picture on the right side of the screen where you have the boxes with total body sodium and total body potassium that are normal, and they are of the same height, meaning that the ratio between sodium and water are within normal limits. Now, when the water in your body changes, that can cause changes in the plasma sodium concentration and plasma tonicity. How so? So when your total body sodium is more than the total body water, okay, then if I would to add water and match the sodium in the body again, then the ratio will be reestablished. So that water that I don't have, that is missing, that is in deficit, is called the free water deficit. And when there is free water deficit, because you have more sodium than water, the plasma sodium concentration will go up. The plasma osmolality will also go up because sodium is the main osmol in the plasma, and the plasma tonicity will go up. Now, when the total body sodium is less than total body water, then now what you're seeing is that you have more water than sodium. And actually, if you look at this red area there, that is the area that is in excess, that is basically that is maintaining this imbalance between sodium and water. So, and this excess water that you have there is called free water excess. And when you have free water excess, because you have more water than sodium, then the plasma sodium concentration is going to go down, the plasma osmolality is going to go down, and the plasma tonicity is going to go down. So, summarizing all of this, we can enunciate as the water dogma. What is the water dogma? Says that the ratio of total body cations to total body water is the main determinant of plasma sodium concentration and plasma tonicity. In total body cations, I'm talking about sodium and potassium. Again, we are going to disregard potassium for practical purposes, and we'll say that the total body sodium, when it's greater than total body water, that means that there is a free water deficit, and that will cause increase in plasma sodium concentration and increase in plasma tonicity. But the total body sodium is less than the total body water, then there is a free water excess, and that will create a decrease in plasma sodium concentration and a decrease in plasma tonicity. How do we assess the state of water in the body? The best way is not by history and physical because they could be unreliable, so the best way is to check a test. And what do you check? You check plasma sodium concentration or plasma osmolarity, which will indirectly tell you what is the state of water in the body. Now, how does the body know that there is too much water or too little water? Do we have water sensors? We don't, but we have sensors that sense the surrogate for water, which is osmolality. And we have what we call osmoreceptors. And we have two classes of osmoreceptors. The central osmoreceptors located in the central nervous system and the peripheral osmoreceptors located in peripheral tissues. The main central osmoreceptor is the organovasculosum lamina terminalis, or OVLT. The OVLT is located in the hypothalamus and is a structure that is part of a family of structures called the circumventricular organs, which are 7 structures in your brain that do not have blood-brain barrier, and therefore they can readily sense any change in the composition of the ECF. How does the process of osmosensation works? Well, if your osmolarity is high, more than 280 to 285, OVLT is going to sense that, and it's going to send a signal first to your thirst centers located in the insula and anterior cingulated cortex, and you're going to be thirsty. The other signal that the OVLT will sense, it will send signals down from the OVLT to another site in the hypothalamus where there are neurons forming nuclei. These nuclei are called paraventricular nuclei, PVN, and supraoptic nuclei. These are neuronal bodies where ADH is produced. ADH will be produced more when the OVLT activates these neurons, and ADH will travel down their axons into the posterior pituitary. The axons are located in the pituitary stalk, so if there is any trauma there, it can cause a damage to the axons and interruption of ADH transport. There is a protein that transports ADH from the neuronal cell bodies down into the pituitary, and it's called neurophysin 2. So then, once it goes to the posterior pituitary, the ADH will be released into the circulation. We also, I mentioned, we have peripheral osmoreceptors that will sense the osmolar content of food, and these are located in portal vein, splenic mesentery, GI tract, pharynx, and esophagus. They're important because certain foods have a high osmolar content and can cause a significant change in osmolality. For instance, soy sauce has a sodium chloride concentration of 16%. If you compare that to what we use for hyponatremia, the hypertonic saline, sodium chloride is 3% only. So there have been actually cases of people who die from just drinking half a bottle of soy sauce because of the severe hyponatremia that it will produce. So when you eat a food that has a high osmolar content, the body will respond in anticipation for a potential change in osmolarity in the same way as if the osmolarity already changed. So it's important for these peripheral osmoreceptors to sense that osmolar content of the foods. And they will stimulate the paraventricular nuclei and supraoptic nuclei, and it will respond by producing more ADH as well. Now, besides having a high plasma osmolarity as the main stimuli for ADH, there is also a nonosmotic stimuli. So there are 2 stimuli for ADH. One is hyperosmolarity, so osmotic stimuli, and the other is a nonosmotic stimuli. So again, we said that the high plasma tonicity or osmolarity will stimulate OVLT, which will increase the production, will send a signal to increase the production of ADH in the paraventricular nucleus of supraoptic nuclei, and then the ADH will travel down to the pituitary stalk into the posterior pituitary where it will be released. A low plasma osmolarity or low plasma tonicity will do the opposite. It will inhibit OVLT, so they will cause no stimuli for ADH production or release into the circulation. Now, the other stimuli for ADH is low effective arterial blood volume, and we mentioned this in the first lecture. When the effective arterial blood volume goes down to more than 15%, then that will stimulate the baroreceptors, which will send signals to the medulla oblongata, specifically a structure called nucleus of tractus solitarius, and then it will send a signal to the paraventricular nuclei, supraoptic nuclei, to produce ADH, and then ADH will be traveling to the posterior pituitary and then released into the circulation. But sometimes these stimuli present at the same time. So for instance, in hypovolemia, you have low plasma tonicity because of the in hypovolemic hyponatremia, you have low plasma tonicity because of the hyponatremia, and you have hypovolemia causing low effective blood volume. So which stimuli is the one that wins? Because low plasma tonicity will cause low ADH, and low volume will cause high ADH. Well, the stimuli for ADH from hypovolemia is much more stronger, because the body always tries to take care of the volume first before of the tonicity, because volume is what's going to kill the person faster. So and that's why I'm making the arrow for low effective blood volume stimulating ADH much thicker, suggesting that the stimuli from hypovolemia is much more stronger than the stimuli from low plasma osmolality. So what is the homeostatic response to hypertonicity? When you have hypertonicity, it's implicit that there is a free water deficit, which is causing high plasma tonicity, and that's going to be stimulating the osmoreceptors, which will stimulate the third center to increase thirst and drink more water. So the osmoreceptors also can send signals to the hypothalamus, which will then increase the production of ADH, and the ADH will travel down into the posterior pituitary, and then it will be released into the circulation. And ADH will work in the kidneys by increasing water retention in the kidneys. So increased water intake and by thirst and increased renal water retention will cause increase in free water, which will improve the initial free water deficit. On the other side, when you have low plasma tonicity or hypotonicity, which is implicit that there is a free water excess there, that will actually be sensed by the osmoreceptors, which will send signals to the third center to decrease the thirst and decrease the water intake. And also it will send signals to the hypothalamus to decrease the production of ADH, which will travel in less amounts to the posterior pituitary, and it will be released less into the circulation, and then the kidneys, because there is less ADH, will retain less water. So low water intake and low renal water retention will cause a decrease in free water in the body, which will hopefully alleviate the excess of free water that initially was seen. Now, how does the ADH work? So ADH works in the kidneys. It works in other organs as well, but we're going to talk about the kidneys. And this is a depiction of a nephron. Part of the nephron is in the cortex, part of it is in the medulla. So we have the glomerulus here, proximal tubule, thin descending limb, thin ascending limb, thick ascending limb, distal convoluted tubule, connecting tubule, and then the collecting duct where ADH works. And three portions: the CCD or cortical collecting duct, the OMCD or outer medullary collecting duct, and the IMCD or inner medullary collecting duct. And this is all of this is the yellow portion is the collecting duct, and this is where water is reabsorbed. And this collecting duct has principal cells where water is reabsorbed, and also there is vessels that are coming from the cortex down into the medulla that are very important. So we'll look at this into more detail. So let's look at what happens here. So the as I mentioned, the principal cells, all the cells responsible for water reabsorption, they're in the collecting duct, and they have two sides. So we have an apical side that is facing the lumen of the nephron in yellow, and they have a basolateral side that is facing the interstitium in yellow, and the vasa recta, which are the capillaries, is called the basolateral side. If you look at the principal cell, inside the cells there are some vesicles that are containing this aquaporin 2 channel that is there. There are no aquaporin 2 channels in the apical side of the cell at this point because there is no ADH. So when water comes, because there is no aquaporin 2, it cannot enter the cell and water is eliminated in the urine. When there is ADH, however, ADH comes from the capillaries and works in the basolateral membrane in the vasopressin 2 receptor or B2 receptor. There are two receptors for vasopressin or ADH. One is the B1 that is in the smooth muscle cells of blood capillaries that their main function is to cause vasoconstriction, hence the name vasopressin, pressing the vessels. And there are B2 receptors that are located in the collecting duct in the principal cells, and it mediates the actions of ADH in the kidneys. So what happens is that the ADH stimulates the B2 receptor, which stimulates a G protein, GS, and this G protein is going to stimulate an enzyme called adenylyl cyclase, which is going to convert ATP into cyclic AMP. So cyclic AMP is the second messenger for ADH. And cyclic AMP is going to stimulate a kinase, which is an enzyme that phosphorylates, and it's called the protein kinase A, PKA. And this protein kinase A is going to phosphorylate serine residues in the aquaporin 2, and it's going to trigger the movement of these vesicles into the apical side, and it's going to insert these aquaporin 2 channels into the apical side. So now water can enter the cell, and then water can come out of the cell through another aquaporins, aquaporins 3 and aquaporin 4, that are located in the basolateral membrane, and they are not dependent on ADH, so they are present there all the time. And then water finally enters the blood vessel to be reabsorbed. Now, you need two things in order to reabsorb water in the kidneys. You need certainly the presence of aquaporin, which is like opening the door for the water to enter, but you also need a stimuli for water to enter. And that stimuli is given by the osmotic gradient produced by the hypertonic medulla. So the medulla of the kidneys, that yellow area there, medullary interstitium, has a lot of solutes. And the main ones are urea and sodium chloride. And the concentration of these solutes at the lowest part of the medulla, closer to the papilla, is up to 1200 milliosmoles. So that hypertonic medulla is the driving force for water to enter. It's like osmotic by osmotic gradient. Okay, so you need two things in order for water to enter the cell. You need aquaporin 2 and you need a hypertonic medulla. So to compare again sodium and water, sort of like summarizing lecture 1 and 2, the excess sodium in your body is going to cause expansion of the ECF and hypervolemia, manifested as edema. The deficit of total body sodium is going to cause contraction of the ECF and hypovolemia. How do we assess the state of total body sodium? By history and physical. We just have to assess what is the ECF volume, and in Didactic will tell us what is the state of total body sodium. What does the body sense? The body doesn't sense sodium directly. It doesn't sense even ECF volume. It senses the effective radial volume. And what are the sensors? The baroreceptors. Okay. And what is the homeostatic response when the total body sodium goes up or down? The RAS system will inactivate or activate depending on what the body needs to do. So the renin-angiotensin-aldosterone system is a hormonal mediator for sodium balance. What about water balance? So the excess of free water in your body will dilute sodium, which can cause hyponatremia and hypotonicity, and the deficit of free water will cause concentration of plasma sodium, so hypernatremia and hypertonicity. How do we assess the state of water? History and physical are not very reliable, so it's just better to send a blood test, and we measure plasma molarity and plasma sodium concentration. So the labs are the best way to assess the state of water. What does the body actually sense? The body actually senses plasma tonicity, which is a surrogate for water. And what are the sensors? The sensors are the osmoreceptors, peripheral and central. And what is the homeostatic response? The body reacts by increasing thirst or ADH when the tonicity is high, or depressing thirst and ADH when the tonicity is low. So these are the main mediators for water handling in the body. So the take-home points are that the ratio of total body cations, sodium and potassium, to total body water is the main determinant of plasma sodium concentration and plasma tonicity. Hypernatremia and hyponatremia are disorders of free water deficit and excess, respectively. And thirst and ADH are the main regulators of water homeostasis. Thank you for your attention.